Good morning, students. This is the sixth full lecture in the Respiratory Physiology chapter. When last we ended, we talked about factors that affect diffusion, this slide here, and either positively or negatively. We talked about equations that needed to be memorized, and we talked about lung capacities and volumes. We also talked about the values, the partial pressures of oxygen, and partial pressures of CO2 that you should memorize that are important for this course and beyond, and a little bit about why there was a decreasing amount of partial pressure as you went from atmospheric to venous, and why there's an increasing amount of CO2 in that same direction. We also mentioned the matching of cardiac output dot Q dot and ventilation, minute ventilation V dot, and how they need to be matched. So you want to make sure that the cardiac output matches the ventilation and vice versa, so that you have enough oxygen coming in and enough CO2 being eliminated to maintain homeostasis. Similarly, the ventilation, the V dot, right? Ventilation, V dot. Remember, whenever you see something like this, it's a rate. Should be matched with your metabolism, right? And remember, the metabolism is all of the biochemical reactions that occur in the body, right? It's how many calories you're burning. So this particular graph measures alveolar partial pressure, right? So in the alveoli, in millimeters of mercury, and alveolar ventilation. And remember, alveolar ventilation is the same as pulmonary ventilation, which is not the same as minute ventilation, right? Because you have to subtract out the dead space, the anatomical dead space ventilation, which is roughly about 150 mils times the frequency of breathing. And what you see here is that depending on where you are, close to sea level, we start at about 150, 160 for total pressure, right? And usually of oxygen. This happens to be CO2. We don't usually get to that level in the atmosphere. So in the alveoli, you have O2 pressure, which is slightly above 100. Now, if you go back here, right, it's about 105 millimeters of mercury, give or take. So these are the curves, right? Normal resting values, right? So this is the peak here. And this is normally where they sit. Usually alveolar pressure is around 100, and normal alveolar CO2 is about 40, which is equilibrated with the partial pressure of arterial blood. As you change, right, as you hypoventilate in this direction, when you hypoventilate, what ends up happening is you move up this curve, so you increase the amount of CO2, right? and you decrease the amount of oxygen, right? Because you're breathing less, therefore you're bringing in less CO2. And because you're breathing less, you're also expiring less CO2. So O2 is going to come in at a slower rate, and CO2 is going to go out at a slower rate. So O2 goes down, right? There's going to be less O2 in the alveoli, and there's going to be an increasing level of CO2. If you hyperventilate, then the CO2 is going to be blown off more quickly. That is going to decrease, and the O2 is going to go up because you're going to bring in more. So the only real way to know this without guessing is you need to have a measure of, typically the way it's done is arterial partial pressures of these gases. Very difficult to do in a human. It can be done. Usually it involves someone who's very, very good at arterial sticks. Usually involves an anesthesiologist. It's dangerous because arterial sticks go into high pressure systems, and there's all kinds of disasters that can happen. But it can be done, but it's very rarely done. So normally we extract blood from humans on the venous side because the blood pressure is very low. That's not going to cut it here. So without knowing that information, you would have to hook somebody up to a metabolic cart, that can measure the expired and inspired amounts of O2 and CO2. And because of this equilibration between alveolar partial pressures and arterial partial pressures, you can get a pretty good idea. But barring some kind of measurement, you don't know that. So the bottom line is, what is this all about? You need to match the two, right? So as one goes up, the other one will go down, and vice versa, right? So this becomes important. When we talk about somebody hyperventilating, what we mean is that usually their CO2 is dropping and their O2 is going up. And when somebody is hypoventilating, the CO2 is increasing and the O2 is decreasing. That's exactly what we mean. If you see somebody breathing more, like in exercise, up to 60% of somebody's maximal oxygen uptake capacity, or their VO2 max, the two match, right? O2 production and expiration, and O2 coming into the system and CO2 leaving the system are matched. And so this number, arterial blood CO2, stays around 40, and that's the number we're looking at. 40 millimeters of mercury for most people is the homeostatic point, and so this direction might be 38, and then this direction might be 42. We're not so concerned about the oxygen when we're looking at hyper or hypoventilation. So here's a quiz of sorts, the effects of various conditions on alveolar gas pressures. So condition, if you're breathing air with low PO2, you have an increase in alveolar vent. So let's go back. If you're breathing air with low PO2, the alveolar PO2 decreases, but the alveolar PCO2 does nothing, right? Because it's just low oxygen air, but the CO2 being produced is the same. If you have an increase in alveolar ventilation, pulmonary ventilation, and unchanged metabolism, what do you think would happen? Take a moment and think about that. You have an increase in alveolar ventilation and you have an unchanged metabolism. It means you're producing the same amount of CO2 as you always have. Well, you get an increase in PO2 because you're bringing in more fresh oxygen. And you're also breathing off more CO2. So the CO2 in the alveoli and in the arterial blood decrease. The next scenario is you have a decrease in alveolar ventilation. Again, the metabolism is unchanged. The amount of CO2 you're producing is the same. So think about that for a moment. Again, a decrease in ventilation, the pulmonary ventilation, alveolar ventilation, and an unchanged metabolism. And if you guess that the alveolar PO2 would decrease and the alveolar PCO2 would increase, you are correct. You're just breathing less, so you're going to bring in less O2 and you're going to get rid of less CO2. Assuming the metabolism hasn't changed, it's pretty straightforward. Now you have a different scenario. The metabolism increases and you don't change ventilation, so the amount you're breathing in and out that is getting to the alveoli, right? Pulmonary ventilation isn't changed, but your metabolism increases. So think about what would happen to alveolar PO2 and alveolar PCO2. And in that case, the alveolar PO2 decreases, and the alveolar PCO2 increases. Look, you're breathing at the same rate. The same total volume is coming in and leaving. Your metabolism goes up. What happens is the cells are now using more oxygen, so that decreases your alveolar PO2, and at the same time you're also producing more waste or more CO2, so you get an increase in alveolar PCO2. Now we have a decrease in metabolism and unchanged alveolar ventilation. Take a moment, think about it. What happens? If you guess that there's an increase in alveolar PO2 and a decrease in CO2, you'd be correct. And for the reverse reasons of the problem above, right? The metabolism slows down. You start to use less O2. Ventilation stays the same. You essentially have an increase in O2. You have a decrease in PCO2 because you're producing less waste. Finally, you get a proportional increase in metabolism and alveolar ventilation. Think about the example I gave you earlier, where somebody is exercising up to 60% of their VO2 max. Think about it for a moment. And there's no change. So you can certainly increase your metabolism and your ventilation. And if you do them in a proportional manner, then you get no change in the PO2, stays at about 100, and no change in PCO2, it stays at about 40. Hopefully that makes sense. You may need to go over that several times. This is a good table to use where you blank out each of these answers. I think making tables like this in physiology for some of the memorization, and maybe you can use some of the Kahoots or some of the flashcards apps online to do this. These are a good way to study by making tables like this. So now let's think about oxygen being carried in the blood. There are two major ways that oxygen is carried in the blood. So don't get overwhelmed by all of these numbers here. The first and the most important way is oxygen is carried by hemoglobin, right? So hemoglobin is a molecule, it's got an iron core, and it's got 4 slots. Every hemoglobin molecule has 4 slots for Essentially, each one can hold one oxygen molecule. 98.5% of oxygen carried in the body is carried bound to hemoglobin. 1.5% is physically dissolved in the blood. Physically dissolved in the blood. How does this relate to the arterial PO2, which equals about 100 millimeters of mercury? Think about it for a moment. Which one of these two do you think determines this? Which one of those two determines that? If you said that it is the 1.5% that is dissolved in the blood, you would be correct, right? So it is the dissolved portion of O2, and it is the dissolved portion of CO2, which we're not talking about in this slide, that determine the partial pressure of that gas in the arterial or the venous system. Whatever is bound has no influence on pressure. Now, when these molecules bound to hemoglobin become unbound, then they can contribute to the partial pressure. In this slide, what we see here is that you can determine in 1 liter of arterial blood, if you have a total of 200 mils of oxygen, based on these percentages, 98.5 and 1.5, 197 mils of that oxygen will be bound to hemoglobin, and 3 mils of that will be bound to, well, won't be bound, will be physically dissolved. If you know that the cardiac output of an average person at rest is 5 liters per minute, and you know that O2 carried to the tissue per minute is 5 liters per minute times 200 mils of O2 per liter, then we can calculate that there's about approximately 1,000 mils of O2 per minute being carried to the tissues. So that's a calculation you should know how to do. This is the hemoglobin molecule with the iron core. That's why iron is so important. Females tend to have lower hematocrits, right? Lower hematocrits, so the number of red blood cells, and they tend to become anemic more than men. So iron, eating enough iron, consuming enough iron is really a big deal for females and rarely is a problem for males. And when you don't have enough iron, you can't make enough functional red blood cells to carry enough oxygen, and therefore there are different types of anemia where you don't have enough red blood cells to carry enough oxygen, and the side effects of that result in people who are anemic feeling tired, right? Because you can't extract enough oxygen, you can't carry enough oxygen. You don't need to memorize this structure. This is just a general picture to show you what hemoglobin looks like. When O2 is bound to hemoglobin, it no longer contributes to the diffusional gradient, therefore it doesn't apply to pressure. And here's a diagram or sets of diagrams that show you this. When PO2 equals PO2 in both chambers, right, and so you have an equal number of molecules on both sides, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6. When PO2 is greater, right, in the blood than in the bound to hemoglobin, then you'll go down its concentration gradient, and as long as there are spaces left on the hemoglobin, oxygen will diffuse to the hemoglobin and will bind. if all of those spaces are taken up by oxygen and the hemoglobin, and what will happen is ultimately you'll have an equal number of free oxygen molecules on this side and a free on this side, so you'll have a new equilibrium. It is only the 2, or in this case the 4 oxygen molecules that are unbound that contribute to the partial pressure. So adding hemoglobin to compartment B substantially increases the total content of oxygen in that compartment, since the bound oxygen is no longer part of the diffusional equilibrium. That's the important take-home message. This is a very important curve. Now, I've done a special mini lecture on this, explaining this. I don't know if in greater detail, but it's specific to this. So if you're having problems with this specific explanation, go to that mini lecture on O2 dissociation curves. So first and foremost, it's another one of those things you need to memorize. You need to be able to replicate this, because understanding this forever as a physiologist, as someone in healthcare, is super important. And so better to memorize it, to have this picture in your head of the O2 dissociation curve. It describes a lot of what happens in the human body when it comes to saturation. You have 2 y-axes. You have a volume percent of oxygen in mils per 100 mils of blood. On the left, you have another y-axis on the right as the percent hemoglobin saturation. Normally in healthcare, you're talking about this. You're rarely talking about this, at least not at the superficial level. And the x-axis is the PO2 in millimeters of mercury. You'll notice that the main curve here, not these two are diseased. The main curve here is a sigmoidal curve, right? So it would do this. As the concentration of oxygen increases, the percentage of hemoglobin saturated with bound oxygen increases, right? So that's what happened. As the percentage goes up, the amount bound on the left goes up also. The percentage of hemoglobin saturated with bound oxygen increases until all of the oxygen binding sites are occupied. That would be 100% saturated. And it's rare that people are at 100%, but they're pretty close, usually between 96 and 100. Note that the PO2 above 100 O2 content does not increase significantly, right? And it doesn't increase significantly above 100 because there's not any place to bind it. So what does all of this mean? Factors which affect binding strength between hemoglobin and O2. The affinity for hemoglobin for oxygen is decreased by an increased hydrogen ion concentration, increased by PCO2, an increase in PCO2, an increase in temperature, and an increase in DPG concentration. So this is called the Bohr effect. The Bohr effect. If you become more acidic, hydrogen ion content goes up, PCO2 goes up, pH goes down, you have an increase in temperature, an increase in DPG. You have hemoglobin, your affinity of hemoglobin for oxygen is decreased. That means the hemoglobin is going to dump the oxygen into the blood at a much faster rate. The best example of the Bohr effect that would contain all of these factors, 1 through 4, is exercise. Under exercise condition, you become slightly acidic, your temperature goes up, and you dump oxygen from the hemoglobin that is bound into the blood so that the cells can use it more quickly. The affinity for hemoglobin for both hydrogen ions and carbon dioxide is decreased by an increase in PO2. That is, deoxyhemoglobin has a greater affinity for hydrogen ions and carbon dioxide than does oxyhemoglobin. So that's called the Haldane effect. So you should know the Bohr effect and the Haldane effect. Here's an example using, now we have it reversed from the last graph. Content is on this side, saturation's on this side. This is again PO2. What you can see here is three curves. When you have the Bohr effect, you have a rightward shift. That rightward shift, right, is the Bohr effect. And you can see what happens here is that if you were to draw a line, you draw a line straight across, you have the Venus saturation at about 75%, right? You have the Venus saturation on this line at about 75%. If you draw a line from each of these straight down, what you'll notice is that when you have a rightward shift, the Bohr shift, this blue curve, you can dump oxygen at a higher partial pressure. So normally you have to go lower. You have to go to a PO2 of 40 to dump the oxygen seen here in this black curve. But you now, if you get a rightward shift, an increase in temperature, an increase in PCO2, an increase in hydrogen ion, a decrease in pH, and an increase in DPG, you can now dump oxygen at a higher pressure. So you can deliver oxygen to the blood, to the cells at a higher pressure. And it's common sense. It makes sense that if you're delivering something at a higher pressure, it's going to get there faster. And under exercise conditions, that's exactly what you need. If you have the reverse occur, if you have a leftward shift, of the oxygen dissociation curve, again, about 75 or so percent saturation, and you drop the curve, you now see that now you have to get to about 30 millimeters of mercury to do this. So the hemoglobin in the leftward shift is holding on to the oxygen more tightly. It's less likely to give it up. And so that would be in the reverse. So all of these would be reversed. You'd have to have this leftward shift, you'd have a decrease in hydrogen ion concentration, a decrease in PCO2, a decrease in temperature, and a decrease in DPG that would cause the reverse of the Bohr effect. Remember that the change in pressure is the driving force for diffusion for this equation. So having a rightward shift is going to cause more diffusion. Anemia and carbon monoxide poisoning. So the bottom line is there are some slight differences here, but for what you guys need to know, both of them severely blunt the total number of spots available in red blood cells to bind oxygen. In carbon monoxide poisoning, carbon monoxide binds about 250 times tighter to hemoglobin than does oxygen. So it outcompetes the oxygen molecules. So there's less spots for oxygen to bind to. So that's why the curve, right? The curve is dropped, the total amount, right? So you, in normal situation, you can get up to 20 mils per deciliter. In this situation, you're about half of the capacity. With anemia, you simply don't have enough iron to make enough hemoglobin. So you don't, once again, you don't have enough spots in the red blood cells, but for a different reason, because you didn't eat enough iron, you can't make healthy cells, you can't carry enough oxygen. So in both cases, the total content of O2 that can be carried in the red blood cells has dropped. How do you fix them? For carbon monoxide, you have to use high concentrations, 100% O2, which will elevate, sorry, I had a graph where we see this here, which will elevate the PO2 to about 600 or 700. And this is going to give you this extra boost here, and that will help you dislodge some of the carbon monoxide, but it's going to take time. So there's a point of no return with carbon monoxide poisoning where there won't be enough time and there's not enough oxygen to drive off the carbon monoxide. So you got to get to people early. With anemia, you have to start eating iron on a consistent basis. You start making new blood cells that have more hemoglobin cores in iron cores in the hemoglobin, and then you can up the carrying capacity. So those are the two take-home messages for that. Now we're going to talk about control of breathing. The regulation of ventilation is a negative feedback loop, the major stimuli for central and peripheral chemoreceptors. You have two sets of chemoreceptors in the body. There's peripheral and there's central. The peripheral chemoreceptors are located in the bifurcation of the carotid arch. In the bifurcation of the carotid arch. So the main carotid splits into the internal and the external. And where that split occurs, you have the baroreceptors. And remember we talked about those with blood pressure regulation. And you have the carotid bodies. The carotid bodies are the main O2 sensors in the body, but they also sense changes in carbon dioxide. So they're stimulated by decreased PO2. So when you go to elevation, you start to become hypoxic. You go to a mile, you go to 10,000 feet, you go to 15,000 feet, you go to 20,000 feet. You become increasingly more hypoxic. These carotid bodies are increasingly stimulated. They cause you to breathe more and more and more because you're trying to bring in more oxygen. The rate matters. If you have an increase in hydrogen ion concentration, which means your pH is dropping, sometimes that is caused by a respiratory acidosis because you have an increased PCO2. These will also stimulate you to breathe more in order to eliminate the acidosis and the CO2. So those are the peripheral chemoreceptors, right? There's a set on each side of your neck where this split happens. The central chemoreceptors are located in multiple places in the brain, including the medulla oblongata, the pons, the hypothalamus, and the cerebellum. They're stimulated by increases in CO2. These are the main CO2 receptors in the body. So when carbon dioxide starts to increase, these are the main drivers of increased breathing. The carotid bodies sense the change in the CO2 first. So I'm trying to get the cursor to where I can use it. The carotid bodies sense the change in CO2 60% faster than the central. But the central chemoreceptors drive 60% of the increase in breathing from the change in the CO2. So they're both important. Here's a look at these carotid bodies. There are also additional carotid bodies. Here's the carotid arches. and there are additional carotid bodies that are attached to the aortics. We think that these are backups in case these sets of carotid bodies in the carotids are taken out for some reason. So they're located in the carotid bodies and the arch. These chemoreceptors, the peripheral chemoreceptors, only on the arterial side of the circulation. They respond primarily to PO2. They does not detect total oxygen content, just the partial pressure, and they provide input to the pontomedullary respiratory network, the carotid sinus nerve and the vagus, and that's what these nerves are here. Ventilation does not change much after small changes in PO2, and that's because the minute ventilation here, V dot, right, V dot over arterial PO2, because most of the curve in the ranges that you're going to see physiologically, right, is flat. And it's not until you get below 60 millimeters of mercury that you're going to have this rapid rise in breathing. So a little bit of a change, actually quite a bit of a change in either direction, does not affect your breathing rate very much. And this also goes back to the shape of this curve, right? You're normally over here. This is the flat part of the curve. You operate really well under all of these pressures here, these percent saturations. So this is flat in the physiological range on the arterial side, and then this is also flat here. So only severe reductions in arterial concentrations of oxygen to blood can stimulate hyperventilation. So when you go to altitude, if you're below 5,000 feet, roughly a mile, you're probably not even going to notice it. You go to 1,000 feet. We're about 600 feet above sea level in the Chicago, Milwaukee area. You go to 800, 1,000, 2,000, 3,000. You're probably not going to notice it much. You get to 4,000 feet, you probably still might not notice it much. So, and that's because of the shape of this curve. So this is another curve that's important to memorize. Here's a diagram of a sequence of events of a negative feedback loop at the peripheral chemoreceptors. This is an interesting way to draw this. It's probably not a bad way. You have a decrease in inspired PO2 because, let's say, you're at altitude. Your alveolar PO2 then decreases. Your arterial PO2 decreases. The peripheral chemoreceptors increase their firing rate via the medullary respiratory neurons. The respiratory muscles contract more. In this case, it's mostly the diaphragm, but the inspiratory intercostals also. And then you have an increase in ventilation, which will then lower, return the alveolar PO2 towards normal, increase it. This is an example of a medulla. Here is spinal cord. Here you have the medulla oblongata. This is the pons, and this is the midbrain. Pons. Right, so this is the brainstem. Obex is the point where the central canal of the spinal cord and the fourth ventricle meet. These are nuclei within the brainstem that you should know that are chemosensitive. The medullary raphe makes all of the serotonin for the brain. Mind you that most of the serotonin in the human body is made by the gut, but in the brain, the medullary raphe makes it. It is chemosensitive, it is highly chemosensitive, so it's sensitive to CO2. NTS has cardiovascular and respiratory functions. It is slightly less sensitive to CO2, but it is still a chemoreceptor. And most structures, nuclei in the brainstem are bilateral. Medullary raphe runs down the midline, so it is not. Pre-Botzinger complex is where the rhythm generator for breathing is located. This is the on switch that triggers the diaphragm to contract. It is also sensitive to CO2. The PFRGRTN area might be one of the most sensitive areas to CO2 that is located close to the pons. You also have CO2 sensors in the lateral hypothalamus and then in the cerebellar fastigial nucleus. The locus coeruleus is one of the least sensitive areas and is located in the pons. So these are all of the chemosensitive nuclei that we are aware of in the brain, and some are more sensitive to CO2 and some are less sensitive. Here's a graph, another one you have to memorize. CO2 effects on breathing. Again, you have minute ventilation on the minute ventilation on the y-axis. In this case, you have the arterial PCO2 on the x-axis. Remember that for most people, arterial PCO2 sits at about 40. Now, unlike the O2 graph, small changes in either direction will have large changes, will have large effects on breathing rate. And that's because CO2, small changes in CO2 affect pH, and pH is one of the most highly regulated physiologic variables in the human body. And because the pH is important for the functioning of all proteins, a small change in PCO2, therefore pH, causes a large change in breathing because you have to highly regulate the pH. O2 isn't as important, and for this reason and this reason alone, CO2 and the level of CO2 in the arteries, again, to reiterate, I've said this before, is the main driver of changes in ventilation in humans. It is unclear if central chemoreceptors sense CO2, pH, or both, so this is a field that both myself and Justin Miller came out of. This is a field we're still associated with, control of breathing, and nobody really knows if it's the carbon dioxide or the pH or the hydrogen ion that triggers these chemoreceptor cells. We just don't know. Tissue, some CO2 converted to carbonic acid via carbonic anhydrase. This is the carbonic anhydrase reaction. So CO2 plus water gives you carbonic acid, and then you could have and this would be the reaction. So regardless of the source, increases in the acidity of blood increases ventilation. And so you can see here that if you take a look at plotting the plasma hydrogen ion concentration in nanomoles per liters, you're normally at about 40, similar to the PCO2, which puts you at a pH of about 7.4, and you have a very robust increase in minute ventilation with relatively small increases in the plasma ion concentration. So again, we don't know if it's the CO2 or the pH that causes this increase in the breathing. So if you produce more non-CO2 acid, it will increase the hydrogen ion concentration in the arteries. The peripheral chemoreceptors increase their firing. The central chemoreceptors will also do that. The respiratory muscles contract more, increase ventilation, which will then decrease alveolar PCO2, which will then decrease arterial PCO2, which will then return the arterial hydrogen ion content back to normal. And on and on. So these trees of physiological responses, which I like using. You might or might not like using them to sort of trace how different changes are connected, are important. I'm not going to go through this. This is much like what your final exam, which is only going to have one major question, sort of a case study, is going to go, where I'm going to ask you to then take all of the systems that we've learned about and show individually, like this, per system, all of the changes that occur, and then try to connect the systems, try to find points at where the cardiovascular system comes in. If we were studying endocrine, which we're not this semester, we would have that. And then we'd have the renal system, right? And so on and so on. The muscular system. Is the muscular system involved? Smooth muscle, cardiac muscle, or skeletal muscle. This one is a really important one to know. The respiratory centers and how it's connected to the motor cortex and skeletal muscle and joints, and how all of these feed into the respiratory center. So your breathing, yes, it's PCO2, arterial PCO2, that drives most of the changes, but the motor cortex, you moving, the cerebellum is involved, joint movement, skeletal muscle movement, temperature, oscillatory changes in PO2, also epinephrine, potassium levels, all of this is involved. So this is a general diagram you should know. And that is it. That is the end of respiratory physiology. Again, this is probably the most challenging section we've needed to go through. I would recommend probably re-watching some of these videos, and certainly watching some of the short videos that I did on specific sections to sort of bone up on these ideas. Remember, there's lots of equations to memorize, lots of diagrams. You got to start putting all the pieces together. I would recommend starting the memorization stuff and doing that a little bit every day, and sort of testing yourself, and on the side also trying to make sure that you understand concepts, right? So you're doing two different things. You're spending part of your studying doing the sort of the easy rote memorization, memorize graphs, memorize equations, know how to use them, and then understanding general concepts. I hope you've enjoyed it. This is really important, especially, again, reminding people that in the future are listening to this during COVID-19. We don't know how long this is going to go on. Some of the predictions are that we may be in this state of never never land for the next year to 2 years, and certainly beyond. There are going to be repercussions. Therefore, understanding this system and physiology for all of you is extremely important, and practical, and understanding some of the things that happen during these types of diseases like COVID-19 and even the flu. So explaining it to your family, and sort of not panicking, and understanding what is being said when we talk about intubation and all of that. So hopefully, again, you've enjoyed this. At least if you haven't enjoyed it, you have a better understanding of the respiratory system and why it is important. So have a great day and study hard.